Hello and welcome to this video podcast for human genetics. In this podcast, we're going to talk about translation and proteins. So let's remind ourselves of the central dogma. Starting with DNA, we go to messenger RNA using transcription. And then we take this messenger RNA and we turn it into proteins. And that's done using translation. Now, in the last podcast, we also added in here, in addition to making the messenger RNA, before it's translated, it gets processed. And we talked about the three ways it gets processed: adding that G-cap, adding the poly-A tail, and splicing out those introns. In this podcast, we're going to take that mature messenger RNA and we're going to translate it into a protein. And then we're going to take that protein and we'll modify it to a mature active protein. So let's go ahead and get started. Okay, so let's go ahead and get started with our discussion of the stages of translation. As we described with transcription, the three stages are the same. We have initiation, elongation, and termination. Not necessarily fancy words, but very descriptive words of what they are accomplishing. Now, to accomplish translation, there's a parts list that we should consider. We need all these things for translation to be successful. So of course we need the messenger RNA. We also need the tRNA. As we said before, it's going to bring in the amino acid as the protein is being made. We need a ribosome. Ribosomes are large structures in the cytoplasm that are made up of proteins and ribosomal RNAs. To make this all work, we also need various protein factors. So these are just various factors or proteins that are needed to allow each of these stages to occur. So we have initiation factors, elongation factors, and terminating factors. Now, building a protein isn't cheap, so we also need energy. Most of that energy will come in the form of GTP, which is similar to ATP that you've heard of in the past. So let's begin with initiation. So I want to draw our messenger RNA. And remember, it's a mature one, so it has that 5' G-cap on the end. And then we have the rest of our messenger RNA with that 3' poly-A tail. Along here are all of those exons that were spliced together. I'm not going to draw exons here because I need to save space as we build up these structures. But these are the exons that resulted after splicing. So when we go back to our parts list, we can cross off the first one. We have the messenger RNA. Now before I go further, I'm going to write the sequence of that messenger RNA that we're carrying forward from our last podcast. All right, so we go back to our parts list, and the next thing we need to bring in for initiation is a tRNA. And for initiation, we need a special kind of tRNA. We need one that's going to bind to our first codon. Now, it's not our first codon because our first codon here is CAC, but this is our initiator codon. Every protein that's being made begins by reading this first codon of an AUG. And this AUG will always bring in a tRNA that has methionine attached to it. How does it know? Well, it knows because this initiator codon will bind to this anticodon here, UAC. UAC is complementary to AUG. This tRNA with this anticodon, in fact, let me write anticodon. This anticodon will always have methionine attached to it. Now, how is it going to find this AUG? I guess there's some low-level chance it could randomly find it, but other items on our parts list for translation are needed. And so part of the ribosome, which we'll call the small ribosome, you will probably be shocked to learn that the other ribosome is the large ribosome. So the small ribosome binds here. Now, by itself, that doesn't accomplish it because we also need various initiation factors and GTP for energy. Small ribosome binds here using initiating factors and GTP. This tRNA is brought into the ribosome, the small ribosome. At this point here, initiation is complete, and we call this structure here the messenger RNA, the small ribosome, the initiating tRNA, We call this the initiation complex. And so now we're ready to think about the second stage of elongation. Now for elongation, the next tRNA needs to bind. And remember that floating all around this ribosome and this RNA in the cytoplasm is every possible tRNA with every possible anticodon. And I should have drawn the anticodon here just to be complete. And so for elongation, our next tRNA has to find its way in here. And our next tRNA is going to have the anticodon of UUU because it's got to be complementary to AAA. We know, and I'll explain later why we know this, but the codon AAA will always bring in a tRNA that has the amino acid lysine attached to it. Just like AUG always brings in a tRNA with methionine attached to it. So this is going to move in here. So now the second tRNA has bound into the ribosomal structures. While it's doing that, around the same time, this large subunit of the ribosome attaches. And this signifies the beginning of elongation, the second tRNA coming in with the binding of the large ribosome. And now the messenger RNA can be fed through this ribosome structure to produce the protein. There are various elongation factors that are important in this process. And I'll call those EF. And of course, we also need energy in the form of GTP. I should also make a comment about the way I've drawn this. You might have come to know I'm not an expert drawer, but I want you to know that all of this occurs inside of the ribosome. Even though the way I've drawn it, it looks like it's poking out of the ribosome. This is all occurring within the safety of the ribosome. So what happens next for elongation to proceed? Well, remember, we're trying to build a protein which is going to have a series of amino acids. So we have to connect these two amino acids somehow. And the way our cells do that is we transfer this methionine over here. So that's what we're going to do. So methionine been transferred over here. And I put the letter N here because I want you to remember that the very beginning of every protein has the amino group at this end. When we get done making the protein, at the very other end of it, it'll be the carboxyl group. So I just kind of want you to keep in mind of where that's at. So the next thing is we need to read this codon and bring in the next amino acid. But we can't yet because the ribosome has these two tRNAs in it. And so what happens is this tRNA is kicked out. So let's go ahead and kick it out. And as it gets kicked out, it gets recycled. It'll get a new methionine placed on it so it can come back and start making another protein. Then, as the messenger RNA is fed through here, the ribosome moves down one. And when it moves down one, this tRNA is now going to be on this part of the ribosome. So I'm going to erase this and show you what it looks like at the next stage. Okay, so with this tRNA at this position of the ribosome, it leaves this spot open. And with that spot open, our next tRNA can come in here and bind to it. And it's going to do so because at its anticodon, it's going to be complementary to this CCU codon. And so the complementary bases here are going to be GGA. We know that the CCU codon will always bring in a tRNA that is proline. Now, the same thing is going to happen. This growing chain here is going to be transferred over to this proline. So let's go ahead and do that. Okay, so it's passed over. This tRNA in this spot of the ribosome is kicked out, and it'll move down one, opening up this UUG codon, which will bring in the next amino acid. Then it'll go to GAG, bring in the next amino acid. It'll keep doing this until it hits a stop codon. Before we get to the stop codon, there's one thing I want to emphasize to you. As these amino acids are being added into this growing chain, I want to talk about what is establishing or making these peptide bonds. Remember, the peptide bonds are the bonds between each amino acid. It's occurring in the ribosome here, but what's the name of that enzyme? And I want you to know this one. It's called a ribozyme. Ribozyme is a generic term. It turns out the specific one in here has a very specific name. But I really want you to walk away from class knowing what a ribozyme is. Ribozymes are RNA enzymes. There's proteins and RNAs in this ribosome, but the molecule that is the catalytic unit that makes these peptide bonds, turns out it's an RNA molecule that functions as an enzyme. This is, at the time they were discovered, it was thought to be rather unusual because we didn't know RNAs could be enzymes. But we now know that they can, and this is just one example. Okay, elongation moves along here. We're making this growing protein chain here, establishing these peptide bonds with this ribozyme. Now it's going to keep doing this. That is, elongation is going to continue until this ribosome hits a stop codon. And so I'm going to redraw this ribosome over a stop codon. Okay, so we finished elongation, and now we're going to talk about termination. So what happens here, the ribosome has been chugging along this messenger RNA, adding amino acid after amino acid after amino acid. And for now, I'm just putting each amino acid as this black dot, just to save room here. But know that we have our methionine, our lysine, and proline, and then the other ones, which we didn't talk about yet. But this is just a string of amino acids. And remember that methionine is always going to be at the end terminus of that protein. Now, at some point, if all goes well, this ribosome will hover over a stop codon. This is one of 3 stop codons. The others are, well, let me just list all of them. UAG, which we see here. UAA is another one. And UGA is the third. You don't need to memorize these. These will be indicated in the genetic code that'll be on your exam, so you don't need to memorize them. Now, the reason it's going to stop here is because there is not a tRNA that looks like this, that has a anticodon on it of an AUC. That would be complementary to the UAG. This does not exist. So what happens when the ribosome runs into a stop codon, it causes a pause. And when it pauses like that, a protein structure called a release factor, we're just going to call it RF, but that stands for release factor, moves into this spot here. And when it moves into this spot here, where the stop codon is, it causes this structure here to disrupt. So when that binds, the ribosome falls off, the messenger RNA is released, and the protein is released. So these all are released from this structure here. And the protein that's released, I'll just draw it like this, with the carboxyl end here and the amino end here. This protein that's been released is now ready to go on to the next stage, which we'll talk about in a moment, because we still need to fold this and we need to activate it. But the protein itself has been made, so we can check that off our list. And we're going to pick this story up here in just a moment here, using this protein here to show the different levels of folding of a protein and how it's activated and how ultimately it is degraded and how sometimes it can lead to disease. But you'll have to wait just a moment for that. Okay, let's move on. Okay, now before we move on and talk about that protein and what it's going to do next, I want to spend a little bit of time talking about the genetic code. The code that was developed to help us understand which amino acid is coded by which codon. So let's talk about that next. Okay, so let's go ahead and discover how we can determine the protein sequence by looking at the RNA sequence. And so I'm going to go back to our previous podcast and write down the RNA sequence we made from that one template strand of the DNA. So let me go and write that down first. Okay, so here's that RNA sequence. And the way we determine the protein from this is we use what is called the genetic code. And this is one from your book. Sometimes this may not appear real big on the podcast, so you may want to pull out your book and look this up. If you don't have your book handy, you can just go on the internet and type in Google image search for genetic code, and you'll find several examples. So as that ribosome is moving down the messenger RNA, usually there's lots of extra sequences before you get to this first one. And so if on a test you're given a large sequence of RNA, what you want to do is you want to look at this sequence until you come to the first AUG. It doesn't matter if there's 20 nucleotides in front of it. You don't do anything until you see an AUG. And once you see that AUG, you know that that is your start. And if you look here on our genetic code, we have an A. Well, let's back up. Let me tell you how you use the code. First letter, second letter, third letter. First letter is A, so you pick up the A here. So that's A. Then the second letter is a U. So you find U along this row here, and it's easy. It's right there. So U. and then you find G with the third letter here, because that's our third letter, G. And so you look in this row here only, you see this G. And then where they meet, we have AUG, and we can see that it brings in a methionine. So our first amino acid, as we already talked about, is methionine. It's always going to be the first. Now the next one is AAA. Pick our A here for our first letter, and then our second letter, A here, so we see where they meet. So AAA is going to be in this block here. And you can see A, and you move over, and there's your A. AAA. We can see that that's going to be lysine. Now on an exam, you will always have a copy of the genetic code. I don't expect you to memorize this. That would be cruel and unusual punishment. Now what I want you to do is I want you to keep reading this as you move down until you get to one of these stop codons. And so I'd like you to hit pause and try to figure out that yourself. And when you hit a stop codon, go ahead and stop and hit play again. And you'll see what the answer is and you match it up. Welcome back. So we had methionine lysine before you hit pause, and then CCU is here, that's proline. Then UUG is here, that's leucine. GAG is here, and that's glutamic acid, GLU. On an exam, you will be giving something probably just like this genetic code, where you'll see the 3-letter code and the amino acid listed out. Now, the next one is UAG, and that's one of our 3 stops. This is a stop, that's a stop, that's a stop. Anytime you see a UAA, a UAG, or a UGA, that's a stop. Now, don't write stop here, because stop is not an amino acid. All you do is stop there. So if you write this on an exam, you'll still get most of the points, but you're going to lose some points for writing stop there, so don't do that. However, this is not quite done. Every protein here, remember, is a series of amino acids. This methionine amino acid has an amino group at the end of it, NH3. And every amino acid, this one, all of them, have a carboxyl in. And so at the end here, we write COO minus, this carboxyl group, to show that we are at the last amino acid. It's like beginning a sentence with a capital letter and ending with a period. Every protein sequence has to have an amino at the beginning and a carboxyl at the end. If on exam you don't put an amino and a carboxyl, you'll still get most of the points, but you will lose some points for not, in essence, using proper punctuation. Now you notice that this messenger RNA has some sequences down here as well as sequences up here that we didn't translate into a part of a protein. And that's fine. That's how it should be. There's always some sequence that is not translated at both ends. So don't think you did something wrong if you end up with extra bases here at either end. You'll definitely have a question like this on the exam, so make sure you understand how to use this table. I also want you to know some features of the genetic code. As we already said, the code is always in a triplet. As you can see here, every amino acid is coded for by a codon, and a codon is this triplet. Again, also, it's the messenger RNA codon. It's not the tRNA, it's the codon here. You should also know that the code is non-overlapping. I should have left my messenger RNA sequence up there, but I forgot. So what we mean by that is that if you have your start codon here, I'm just going to make up some codons here. So if this was our messenger RNA, when I say it's non-overlapping, what I mean is this, that this makes methionine, this makes threonine, this makes serine, and this makes glycine. Non-overlapping means that it only reads 3 codons at a time. We don't read it this way, AUG, and then UGA, and then GAC, and so forth. We read it 3 base codons at a time, 3, 3, 3, 3. So we don't do this. The genetic code identifies the start and stop of a protein. In fact, I'd rather say end of a protein. So it identifies the start and end of a protein. The start is identified by the methionine always, and the stop is identified with one of 3 stop codons. You should know that the code is universal. And what we mean by that is all life uses this code. Bacteria use this code, fruit flies use this code, a tulip uses this code, fish use this code, humans use this code. It's universal. And we say the code is degenerate, which sounds like a bad thing. I hate this word, but that's what we call it. And we call it degenerate. I think it's part of the history of how it was discovered. It was discovered by physicists, and physicists use this word degenerate in a way different than we use this word in society. And what we mean by this is that there are redundancies in the code. And what I mean by that is, for instance, there are 2 codons here that both make histidine. There are 4 that make threonine. There are these 4 plus these 2 up here that make leucine. Most of the 20 amino acids can be made by more than 1 codon. So we say it's degenerate or it's redundant. Some of them don't. For instance, methionine, there's only 1 code for it. Tryptophan, there's only 1 codon for it. But there's a lot of degeneracy or redundancy within the code. Okay, that's all I want to say about the code. All right, now what I'd like to do is talk about protein folding, processing, and disease. If you remember from a whiteboard ago, or maybe 2 whiteboards ago, we ended with this protein and we just signified it with these linked amino acids, with the amino end here and the carboxyl end here. This linear sequence here is called the primary level. It's just a linear sequence here, and we should say that this linear sequence is non-functional. A functional protein has to move beyond this linear sequence. The next level we call secondary. And this secondary level has this protein folding upon itself in regions near it. And I'm going to show this by showing 2 common ways that the secondary level folds. And the first is what we call an alpha helices, or alpha helix. The other is something we call a beta sheet. These occur because there are bondings, hydrogen bondings. I won't ask hydrogen bondings on the test, but it is hydrogen bondings between this region and this region that bind to each other to create these secondary level of structure. And we're going to call these, for the most part, non-functional. There's an important exception to this that we'll talk about in a moment. But usually a protein needs to move beyond the secondary level to the tertiary level. And what happens in the tertiary level is that the various secondary level structures will fold upon each other. And so I'm going to draw something like this, where you have alpha helices, beta sheets, an alpha helix, and an alpha helix. They begin to collapse upon each other. And the important thing about this level of folding is that it's kind of hard to predict how it's going to fold, right? Because you could have this region of the protein in the primary level, when it folds upon itself like this, may actually be interacting with this amino acid. This is secondary structure is more local areas of binding to create these alpha helices and beta sheets. But the tertiary takes advantage of features on all ends of the protein to form this kind of big globular structure of the tertiary level. Now this form is functional. Sometimes we have to do things to it, which we'll talk about in a moment, to make it functional, but in general, this is the level of folding that we think of as a functional protein. Now, there's one last one called the quaternary structure. The quaternary level, I'm just going to use this one up here, is when you have one protein, so this is, let's just call it protein A, it binds to a different protein. And so I'm going to draw another tertiary protein here, and we'll call it protein B. These two will interact with each other by various mechanisms to form a larger protein composed of 2 separate proteins. So tertiary level is a single protein. Quaternary level is 2 proteins bound to each other. Not all proteins have a quaternary level, but if they do, it's the quaternary level that is functional. So what I want to talk about is this special protein called a chaperonin. And so I'm going to draw our linear protein here, our primary structure. And so that's the symbol for primary. This is our primary structure. I'm just drawing it this time as a straight line. What happens is, as this is being released from the ribosome, we have these proteins called chaperones that can bind to it in various ways. And when they bind to it, it allows the most stable conformation of this protein to form. So as it goes from primary to secondary to tertiary and maybe to the quaternary structure, where we have two proteins, which I'll just symbolize like that, coming together and binding in this final quaternary structure. These chaperonin proteins are incredibly important in making sure that the optimal way that this protein can fold, it will fold that way. Without the chaperones, sometimes proteins won't form correctly, and they will fold in an incorrect formation. So chaperonins are proteins that allow for the most stable protein formation. Now, I alluded to something on the previous whiteboard when I showed you this tertiary structure. And I said it's usually active at this level, but sometimes it needs to be processed or modified in a certain way. And so I want to talk about that real quick here. Sometimes we have to add molecules to this protein to make it functional. And one way that we can do that is by adding phosphate groups. So I'm going to put phosphorylation here. And that's just adding a phosphate group. So you may still have this protein forming, but adding this negatively charged phosphate group may allow this protein to fold a slightly different way, or may allow it to interact with a different protein in a more efficient way. But in this example here, this would be inactive and this would be active. There are other ways we can activate a protein, and we're just not going to talk about those. But one way, and one really important way, that we see lots of examples of in humans is by adding a phosphate group to that protein to go from inactive to active. All right, I do want to talk about one other way we can modify it, but it's not necessarily to activate it. It's sort of the reverse, and we're going to call this ubiquitination. And what we're doing here is we're adding what we call a ubiquitin group to it. In fact, when we do this to our proteins, we typically add a whole bunch of these ubiquitin groups. So this protein here, at some point, the cell may decide it needs to get rid of it. Maybe it's misfolded and it's not working properly. Maybe the cell wants to get rid of this protein because the job that it was doing is no longer needed. For various reasons, we need to get rid of proteins, and ubiquitination is one of those. So when we add ubiquitin to a protein, that targets it for destruction. And I should also state that when this is degraded, it's not a complete waste, because what's released here are all of those amino acids. And these amino acids can be fed back into translation to make new proteins. It wouldn't have to make the same protein because it doesn't care. It just is going to be used into making any new protein that needs to be made. The cell turns out to be an expert in recycling. It reuses these amino acids from this degraded protein. So I want to talk about next this last part here of disease. And we know that misfolded proteins can lead to disease. Sometimes a protein misfolds because of some kind of environmental condition. Temperature, pH, these different things can change it. But that's not what I want to talk about here. But that can happen. I just don't want to talk about that in this example. What I want to talk about are inherited diseases. When you have a mutation that changes how the protein folds. The first example I want to talk about is this protein called CFTR. It's the one linked to cystic fibrosis. And so here's our cell. And we know that we have this nucleus, we have an ER here, and we have these Golgi stacks. And you don't have to worry about this in this level of detail, it's just helpful in explaining it. We know usually what happens is that this CFTR protein here is made in the cytoplasm, or at least it started to make in the cytoplasm, and it goes into the ER, and then it gets shuttled into the Golgi, it goes between the different Golgis, and ultimately it's placed inside of a vesicle, and it's taken to the outside of the cell where it forms this channel. At the plasma membrane here, it will help regulate salt balance. It'll help maintain the homeostatic nature of the cell. Now, people with a mutation in this gene, what happens is the protein never makes it here. The protein begins its synthesis and is in the ER, and in the ER, it misfolds. And instead of being transferred to the Golgi, it gets pushed out of the ER into the cytoplasm. It's misfolded, it's in the cytoplasm. It doesn't go through this process where it has to go in order to fold properly to be in the plasma membrane to be a channel protein. So in this cytoplasm, what happens is it gets ubiquitinated and it gets degraded and it never makes it to the outside here. Different forms of cystic fibrosis. This is just one of them. So misfolded protein leads to this disease of cystic fibrosis. I do want to spend the last few moments talking about a prion disease. This has many different forms of it. Some that you may have heard of would be like mad cow disease, chronic wasting syndrome found in deers, CJD disease found in humans. And prion diseases are very interesting. They're characterized as being infectious protein diseases. In essence, what happens here is you have a protein that is primarily composed of alpha helices. And like with the cystic fibrosis example, these proteins end up on the plasma membrane. When they begin to misfold, sometimes for reasons we don't completely understand, these alpha helices are converted into predominantly beta sheets. In this form, what happens is on the cell, these begin to aggregate and they're not allowed to do their normal function. This ultimately can lead to cell death of the neurons. And that's why someone with CJD disease or a cow or a deer with these diseases, when you examine their brains, it appears as if there's a bunch of holes in it because these proteins, by aggregating, have destroyed parts of the brain. Now, why do we say it's infectious? This is the thing that's a little hard to understand. Once one of these misfolds, it will find other properly folded ones and convert them into the misfolded ones. And as this happens, each misfolded one now will go on other correctly folded proteins into misfolded proteins. So that's why we say it's infectious. It's not infectious in the sense that we think about a bacterial infection or a virus infection. It's infectious because the protein itself converts other proteins to misfold. The unfortunate problem with prion diseases, like CJD, is that there's no cure for them. There's no cure for mad cow disease or chronic wasting syndrome. And you can acquire this disease by eating infected meat from cows or infected meat from deer. If you happen to have meat, like say you had a deer that you suspected had chronic wasting syndrome, you immediately need to contact the DNR so they can properly dispose of it. I once had somebody tell me that if you cooked it long enough, you would kill this problem. No, you can't do that. You cannot get rid of it. You have to call the DNR and have them properly dispose of such a deer. If you eat it, you run a risk of developing one of these prion diseases, and nobody wants that. Okay, that ends this podcast. In this podcast, we picked up from the last podcast with our messenger RNA, and we showed how we converted that into a protein. And then we talked about how that protein could be folded and activated, and how sometimes when proteins misfold, they can cause disease. If you have any questions at all, please make sure you ask me. If not, I will see you on the next podcast. Bye for now.